COVID vaccine varante kutta.
Good afternoon, friends. On behalf
Good afternoon, ma'am.
On behalf of the Nutrition Society of India Mumbai chapter, myself, Dr. Subhadra Mandalika, extend a warm welcome to each one of you to today's much awaited webinar on one of the most interesting topics, chrononutrition. As many of you must be aware of, our ancient scriptures have given us certain rules regarding food consumption. According to Manu Samhita, we should avoid eating before sunrise and after sunset. The Ahar Niyam of Ayurveda says, eat breakfast moderately, lunch well, and eat dinner very sparingly. The modern verse of this same is, eat breakfast like a king, lunch like a prince, and dinner like a pauper. There are many ways in which the same wordings have been expressed again and again. But the big question is, how many of us actually follow this? Majority of the people do not follow these rules given to us by the ancient scriptures only with one reason, that is, we are not getting the kind of scientific evidence that we demand from these scriptures. Today, we are with you with an eminent expert who is ready to share a very strong scientific evidence on the body clock, the circadian rhythms, and how the food consumption and thereby the health status of an individual changes according to the same. Let me first introduce our eminent speaker, Dr. Johnston, Dr. Jonathan Johnston. Dr. Johnston is a reader in Chronobiology, Integrative Physiology at University of Surrey, UK. Besides holding this responsibility, Dr. Johnston is also a postgraduate research director of School of Biosciences and Medicine. He is a member, School Athena Swan Self-Assessment Group, and he is also a member, Faculty Research Degrees Committee. His main research interest is in biological rhythms, the most apt interest, which is going to give us a lot of valuable information on circadian rhythms. And he has coordinated major research projects, and one which is very close to today's topic is the Big Breakfast Study, Chrononutrition Influence on Energy Expenditure and Body Weight. He has several achievements through his research endeavors, and the most notable ones are listed over here. And these achievements reveal to you that how he has developed valuable methodology to assess the circadian rhythms and the body's response at various times to food consumption and the resulting changes in the body composition. And he also tried to relate it to the food consumption practices. He has around 54 research publications, which are very valuable because most of them are revolving around chrononutrition and in highly reputed international journals. He has been an invited speaker at various national and international conferences. Not only that, the most valuable output or contribution of a scientist is his contribution not only to the fellow scientific community, but to the community at large. So his research, Dr. Johnston has shared through the media, through various articles, as well as interviews on podcast and video interviews. You can see that these topics are so very relevant to you and me and everybody. He has affiliations and memberships in various reputed organizations. Today, Dr. Johnston is going to speak to us on role of chrononutrition in metabolic health. With these few words, I would like to invite Dr. Johnston to take over and start his presentation. Dr. Johnston, over to you.
Well, thank you very much indeed for that, that kind introduction. I'm very happy to be here and I look forward to sharing some interesting science with you. And more importantly, I look forward to answering any questions that you have at the end of the presentation. So let me begin by giving you a very brief outline in the talk that I'm going to give you today. Just to set the scene, before I start talking about nutrition in more detail, I just want to give you a very brief overview of the science of circadian rhythms and our circadian system. So this is the system that we have in our bodies that regulates internal daily rhythms of physiology and behaviour. So this will hopefully only take 5 minutes or so. I then want to talk a little bit about so-called peripheral clocks. A lot of people know that we have a so-called master clock within our brain, but as we've found out over the past 20 years or so, we also have clocks throughout our entire bodies. And basically every single tissue in our body, possibly every single cell in our body, actually has its own clock. And recent work, particularly from animal studies, but there's also mounting evidence in human research, shows that these peripheral clocks that we find in tissues such as our liver, our pancreas, our intestines, are actually very, very important for our overall metabolic health and biology. So that again will be fairly brief to set the scene. What I then want to spend most of the time talking about is how we can regulate our circadian rhythms, our body weight and our metabolic health simply by changing the timing of our meals. So this will be about half of the talk, and I'll break it down into different sections depending upon different ways in which we can use meal timing. And then finally, for those of you who are working in clinical practice, right at the end, I've just got a little slide or two just to bring it all together and to summarize what I think is good advice that we have based on science that you can use in your clinical practice. And we can talk about areas which are well studied and areas which perhaps need a little bit more evidence before we can promote them strongly to the public. Where necessary, I'll mention a little bit about some of the basic animal and cell biology that's been done, but in the vast majority of cases, I'm going to focus on human physiological studies, as I think that's most relevant to most people here today. So, daily rhythms in humans. I'm sure we're aware that many aspects of our behaviour and our physiology change across the day. For example, we know that sleepiness, our tendency to want to go to sleep, changes across the day. Another big factor in research at the moment is our daily rhythms of metabolism. And clearly that's by far the most relevant human change that we're going to talk about today. But there are many other aspects as well that are worth mentioning briefly. Our mental performance. During our biological day, we are much more alert. Our cognitive ability is much higher than, for example, it is during the biological evening and the biological night. We also have fundamental changes in our core body temperature. Most people will get about 0.5 to 1 degree Celsius change in their core body temperature over the day, with it dipping during the night. Many of our hormones are secreted according to a strict daily pattern. The most obvious example here is a hormone called melatonin, which you may have heard of. Other things such as blood pressure, the occurrence of disease symptoms and mortality have also had very strong daily rhythms reported. So very briefly, what terminology do we use to describe these? Well, of course, in normal life, we are not only having our rhythms within our internal biology, but we also have rhythms in our environment. So very simply, the sun goes up during the day and it's down during the night. We get daily changes in our temperature. We change our body posture. We're more active during the day and hopefully we're resting at night. And these rhythms are referred to as diurnal rhythms. So they are a combination of our internal biology, our internal clock system, but also our changes in environment and behavior. So these are ones which are most directly related to our normal life. But of course, it's difficult from a scientific point of view to say where these rhythms come from, because there are so many influences both within and outside of our own bodies. So if we want to study true internal biological rhythms, and these are what we call circadian rhythms. This is very nice to do from a mechanistic biology perspective, but of course it's very difficult to do, and it deliberately removes some of our external factors. However, when people have done these experiments, what we are able to show in animals, but also very clearly in humans, is that we do have these internally driven rhythms. But we need specialist laboratory protocols and methods to be able to reveal them and unmask them from the environmental cues. So very briefly, here we have one example of a laboratory protocol which we can use to actually unmask and reveal our true internal biological rhythms with no external influence. And I'm only mentioning this because it will crop up a little bit later on within the talk. So one of the ways in which we can do this in the laboratory is to use what we call a constant routine protocol. And this is designed to remove the effects of the external environment, but also behaviors such as sleep. So in one of these cases, we have volunteers coming into our laboratories for about 40 hours. During this time, they will have no knowledge of the clock time. They will be kept in constant dim light. So there's enough that they can see, but very little more than that. And there will be no change in that light exposure across the 40 hours. They will be kept virtually lying down and not allowed to move from that position. So we control their posture, so they're not getting up and moving around. We keep them awake to remove the daily sleep-wake cycle, and we avoid any feed-fast cycle by giving them small identical snacks every hour across this protocol. So by doing so, we're removing the light-dark cycle, we're removing the sleep-wake cycle, we're removing the feeding-fasting cycle. And by doing this, any rhythms that we see must be purely down to internal biology. And when we do these sorts of studies, again, we can reveal all sorts of different rhythms. So this is a diagram which shows you the sorts of rhythms that human beings exhibit when their behavior is matched to their environment. So this is, if you like, in a healthy, normal state. So in the middle, we have our person here, and we have these clocks throughout the body, within the brain, but also in tissues elsewhere in the body. In a normal, healthy environment, light is perceived during the daytime, and of course, it's dark during the night. And the feed-fast cycle is such that people are eating typically during the day and fasting during the night. And we see various rhythms. Here are some simplified data shown for you. So on the top left, we can see an example endocrine rhythm, and this is the rhythm of the hormone melatonin. And you can see it's very low during the day, and you get this large increase in the evening, and which comes down again during the following morning. At the bottom left of the slide, we can see the example of core body temperature. And here you can see nicely how core body temperature drops by about 0.5 to 1 degree Celsius during the biological night. We also see changes in energy expenditure. And on the top right of this slide is a cartoon representation of a paper that was only published a few years ago. And this is showing resting metabolic rate. And even our basic resting metabolic rate changes across the day and is thought to dip by about 10% during the biological night. We can also see changes in metabolites, and again, I'll come back to this later on. The bottom right of the slide here just shows you one of the clearest examples, and this is looking at triglycerides, so the most common blood fat molecule. And if you measure triglycerides across the day, you will find very reproducibly that the basal concentration without any meal will actually rise by about 50% during the nighttime compared to during the day. So these are some of your, if you like, natural rhythms in a well-aligned state. However, it's also worth bearing in mind that particularly in the modern world, where more people are working late in the evening or possibly doing night shift, that a lot of people are not in what we would call a natural, healthy, aligned state. So on the right of this slide here is a cartoon representation of what, unfortunately, more and more people are suffering from. So people have light exposure during the night. Since the invention of the light bulb, of course, we have much more control over our lighting environment. We can switch lights on and off when we want to, not just be dictated to by sunrise and sunset. People are often also eating late at night, and to compound the effect of that, it's been shown that when people eat at night and when they're tired, they tend to choose higher energy dense foods compared to the healthier foods they might eat normally during the day. And this has two effects. Light exposure and feeding time during the night can disrupt the circadian system. And you can see these warped clock images on the cartoon here representing the disrupted circadian system. But it can also have effects on health. And again, I'll give you some examples of eating, particularly at the wrong time of day, and how that affects metabolic health later in the talk. So to summarize this first part of the talk, many, probably most aspects of our biology as human beings changes across the day. In the laboratory, we can use highly specialized methods to reveal our, if you like, pure internal biological rhythms. However, in real life, these rhythms that do exist are also influenced by the environment, particularly things like the light-dark cycle, our feeding-fasting cycle, and our behavioral cycle. And if we have mistimed behavior in relation to our normal body clock system, such as light and feeding at night, but also people engaging in regular shift work or regular jet lag, this can disrupt our circadian system and can be bad for our health. So that's really the take-home message for this first part of the talk. What I want to now move on to is to introduce you to some of these rhythms that we have in metabolism, which are driven by clocks throughout our body, and to hopefully convince you that these are very important. And due to tools that we've recently been developing, some at Surrey, but many around the world, we are now able, for the first time, to actually measure all sorts of metabolic rhythms within humans, not just rely on animal studies. This is just the only animal slide that I'm going to give you in this part of the talk. And this is a study that was published 10 years ago now by Joe Bass's group in the United States. And this is one of many studies that have been published that use a type of genetically modified animal and give a very clear indication that clocks in particular parts of the body are very important for our whole body metabolism. So in this case, they used a mouse which had a dysfunctional clock within its pancreas, but everything else in the body was perfectly normal. The only thing that was different was it didn't have a functional clock within its pancreas. And if we look at the graph at the top right of this slide, here we can see one of the metabolic physiological tests that they did on these mice. There are two solid lines here, shown by the hatched and the white circles. These are the control animals, and what you're looking at on this graph here is what we call a glucose tolerance test. So at time 0, animals were injected with a controlled amount of glucose, and you can see in the control animals there was a peak in the blood glucose concentration about 30 minutes after the injection, and this came back down towards baseline an hour or two later on. The interesting part is the dashed line that you see at the top of the graph. These are the data that come from the animals that lack a functional clock within the pancreas. But again, remember, the rest of the animals are completely normal. When they don't have a clock in the pancreas, the same insulin injection leads to a much higher concentration of glucose in the blood, and it takes a lot longer for that glucose to actually come back down to basal levels. So even 2 hours after the glucose injection in these genetically modified animals, the glucose in the blood is still higher than the peak concentrations that you found within the control animals. So there's a profound inability of these animals to deal with glucose. So they are intolerant to glucose. And the short answer as to why this happens is that when you disrupt the clock within the pancreas becomes unable to secrete a normal amount of insulin in response to a high glucose concentration. And this has been shown now by multiple groups doing similar studies within the pancreas, but similar results have been found when you knock out the clock in the liver, in skeletal muscle, in white fat tissue, and a whole range of other cell types. So these clocks within our peripheral metabolic tissues are extremely important for our metabolic health. Okay, so let's move back on to humans. So one problem that those of us studying human circadian rhythms have had historically in the field is that for obvious technical and ethical reasons, we are limited in how we can study metabolism in humans. But one breakthrough that came now nearly 20 years ago was the discovery that if you take regular blood samples from participants, one of the few tissues that we can obviously ethically take, you can actually isolate the white blood cells, and if you look at gene expression within these blood cells, you see beautiful circadian rhythms of gene expression. It doesn't matter what these genes are, but hopefully what you can appreciate from this slide is that all of these genes in 3 different participants along the top, middle, and bottom of this slide are all showing beautiful rhythms with a 24-hour periodicity. So these are circadian rhythms within white blood cells from humans. That's very nice, but of course, if we're interested in metabolism, we would like to be able to measure things that are a bit more directly related to metabolism than white blood cells. And there are various ways in which we can do that. There's a technique called metabolomics, which has been fairly recently developed in the bioscience literature. And very simply, this is a way in which we can measure many hundreds, or in some cases thousands, of metabolites in a single small sample from humans or any other source. And this was first used in humans on a circadian basis now in 2012, so 8 years ago. And the basic take-home message is that when people have sampled tissues such as blood, but also saliva and urine from humans, when you measure hundreds and hundreds of metabolites within these samples, you find that between 15 to 20%, so nearly 1 in 5 of all metabolites detected actually have a pronounced circadian rhythm. So even in metabolites circulating around our body, so many of them, hundreds of them, thousands of them, are actually rhythmic across the day. And if you look at what these rhythmic metabolites are, they fall into typical classes. And I've listed some of them here on the bottom left of the slide. So some of them are called acyl carnitines. These are very important in lipid metabolism, particularly in the mitochondria within cells. Amino acids, which of course are very, very important in protein biology. Phospholipids, which are important in cell membranes. And another group called sphingolipids, which are important in cell signaling processes. So these are really bioactive metabolites, which are showing rhythms within our circulating blood. What can we do, though, if we want to move beyond blood and actually look at the metabolic tissues themselves? Well, I like this slide because it's from my first ever human study at Surrey. And what we did simply here was we got volunteers in, and every 6 hours, we took a small biopsy of white fat from them, from an area that we know is metabolically active. And in this first study, we just did some very simple gene expression. It doesn't matter, what these genes are, but hopefully what you can see is that there are beautiful 24-hour rhythms of gene expression in biopsies of human fat taken across the day. And we've recently expanded that analysis, and here we've used a method which enables us to measure every single gene. So all the 40,000 genes that the human body has, we were measuring. And we found that about 2%, so that was about 850 genes that we found to be expressed in human fat, actually showed a 24-hour rhythm. So hundreds and hundreds of genes within human fat are circadian, are going up and down across the day. We then looked at what the known functions of these genes were, and they were involved in core metabolic processes within the fat, such as lipid metabolism, DNA and RNA metabolism, and also redox activity. So these are key metabolic functions of fat tissue, which are changing across the day. And then finally, in collaboration with Charna Dibner's group in Switzerland, but many others, we did a similar study looking at human muscle, and human muscle biopsies were taken 4 hours apart across the day. And again, we found many hundreds, I believe thousands of genes that showed daily rhythms. And these were involved in key metabolic parts of muscle tissue, such as glucose homeostasis, lipid metabolism, and the secretion of myokines, which are muscle-derived hormones. So to summarize this second introductory part of the talk, we have clocks throughout the body. The main clock in the region of the brain is known to be what we sometimes refer to as the master clock, and is the best known. But please remember that we have clocks all throughout our bodies, including in all the key metabolic tissues. And studies, particularly from animal experiments, have given us very clear evidence that these individual clocks in our metabolic tissues are extremely important for whole body metabolism. And the good news for those of us interested in human biology and treating patients is that we have recently, over the past 10 years or so, developed a range of new methods, which are still in ongoing development, which are now enabling us to study the basic biology of metabolic rhythms within human studies. And of course, we can now start to apply these new methods to more applied situations, such as nutritional interventions. And that's really what I want to go on to now for the majority of my talk. So I'm now going to move on to how we can use meal timing as a tool to regulate various aspects of our physiology. I'm going to start off by showing you an experiment that we published a few years ago that shows that meal timing can actually be useful in resetting and synchronizing some, but not all, of our internal circadian rhythms. And so this may, in the future, enable us to use meal timing as part of a treatment to help people who are suffering from jet lag and possibly improve the health of people like shift workers as well. I'll then introduce you to some example data showing you how the post-prandial response, the response over a few hours and how we process meals actually changes across the day. And this is very important because it can show you how, for example, shift workers have had high incidence of metabolic diseases. And part of the reason for that may be that they're eating during, if you like, the biological night. And this is actually having an impaired effect on their metabolic responses. Then the final two parts of this section that I want to talk about are longer term interventions where we can use meal timing to actually improve potentially body weight and long-term markers of metabolic health. And I imagine that for many of you, this is going to be the real crux of the talk. And hopefully the basic science I've just explained to you will hopefully set up the rationale for why these studies have been done. But let me first of all talk about how we can use meal timing as a tool to help reset our clocks. And this was first shown in mouse studies. In this case, it was 20 years ago by a group in Switzerland. And they did a very simple experiment. They had animals on a 12-hour light, 12-hour dark cycle. Half of the animals had exposure to meals, or they had food availability rather, when the lights were switched on, but not when they were switched off. The other group had the opposite. So they had food exposure when the lights were off, but not when the lights were on. And the basic take-home message here is that the so-called master clock within our brain remains synchronized to the lighting environment, irrespective of when food was given. However, the clock within the liver of these animals synchronized perfectly to the meal timing. So when food was given was the dominant synchronizing factor for these liver clocks, and the food timing actually pulled the liver clocks away from the light-dark cycle and aligned them perfectly to the meal timing. So at least in animal studies and for some tissues within the body, we know that food timing is an incredibly powerful way of synchronizing clocks. But does this translate to humans? doesn't necessarily mean that if we see it in animals, we'll see it in humans. And so this was a study that we published about 3 years ago now. And this is a long-term laboratory study. And this is a summary of our design. So I won't go into too much detail, but basically a week before the participants came into our lab, we had them on a fixed light-dark cycle and a fixed sleep-wake cycle and a fixed mealtime cycle. So we asked them to get out of bed at about 7 o'clock in the morning, have their breakfast about half an hour after getting up. They then had a lunch half an hour after their breakfast and a dinner half an hour after their lunch. And they did this for a week before coming into the lab to try and synchronize their rhythms as well as possible before they actually came to the university. And when they came to the lab, they then lived in our laboratory for 2 weeks. So this is a very, very long, intensive study with our participants not leaving the laboratory for 2 weeks. And for the first 3 days of the laboratory study, they had the same basic behavioral cycle as they had before they came in. So they got up at about 7, they had an early breakfast, then the lunch around about midday, and the dinner around about 5 or 6 p.m. We then see this grey area on the chart here. This means that we then put them through one of our constant routine protocols to assess their internal circadian rhythms. So that lasted for 37 hours. We then gave them a night off so they could actually have a bit of recovery sleep. And then for the next 6 days, they had exactly the same light-dark schedule and exactly the same sleep-wake cycle. The only difference was each of their 3 meals was delayed by 5 hours. They then had another constant routine protocol towards the end of the study. And so we were able to measure their internal rhythms after the early meals and after the late meals and compare the two. And we looked at various different parameters. Here are just some subjective questionnaire scores that we had. So on the left of the graph, we have a well-validated sleepiness scale. And as you would expect, the longer that these participants were in their constant routine, the more sleepy they got. Remember, they weren't allowed to sleep during this routine. And so what this shows us is the beautiful circadian rhythm of sleepiness that we see. These individuals were awake for the whole 37 hours. They became more tired during the biological night, but when they came into the biological day, they actually started to feel more relatively awake again. So this is a lovely way in which you can show that circadian rhythms influence sleepiness. But there was no difference between the groups. So the early or late food had no effect on the subjective rhythm of sleepiness. We also looked at subjective hunger, and as others have shown, there is actually a daily rhythm of subjective hunger, and people tend to feel a little bit more hungry during the evening, which then drops off over the night and into the morning. And we were able to reproduce that basic rhythm, but again, we found no difference in subjective hunger scores after the early or the late meals. We then looked at hormonal markers of the so-called master clock within the brain. And here you're looking at the left, plasma melatonin concentration, on the right, plasma cortisol concentration. And as we would expect, both of these hormones show beautiful circadian rhythms, but there was no difference, again, caused by meal timing. And that was what we predicted from the animal studies. However, where things got interesting was where we looked at metabolic markers within the blood. So on the left of this slide, we are looking at plasma glucose concentration. And remember, these are individuals who are being given a very small identical snack every hour. So there's no big meal being given to these participants at all. And as others have shown, there is a beautiful circadian rhythm of plasma glucose concentration if you remove big meals from these individuals. The solid line represents the glucose rhythm after the early meals, and the dotted line represents the glucose rhythm after the late meals. And you can see that by delaying meals by about 5 hours, that actually affects and delays the internal biological rhythm of glucose by about 5 to 6 hours as well. So this shows that the plasma glucose rhythm, the rhythm of glucose homeostasis in our bodies, is actually profoundly regulated by meal timing, and meal timing is actually the dominant time cue over things like the light-dark cycle. So glucose homeostasis and the rhythm in glucose homeostasis is very profoundly regulated by meal timing within humans. Interestingly, we saw very little change in plasma insulin concentration. And so the glucose rhythms may be due to things like a change in insulin sensitivity across the day rather than insulin secretion. We also found no difference in plasma triglycerides. And so here, although we're seeing a big change in the rhythm of glucose, there's no change in the rhythm of plasma triglyceride. And this might seem a little bit surprising. Why is one sensitive to meal timing and one not? Well, there is actually, again, evidence from other studies in animals and also humans that the rhythm of triglycerides within the blood is actually mainly driven by our master clock in the brain and not by our peripheral clocks. And so that might explain why glucose rhythms are reset by food, but not triglyceride rhythms. Very briefly, we also looked at gene expression within biopsies. We didn't see a big change here. We saw a delay of about 1 hour in these different gene expression rhythms within our fat biopsies. So not quite as impressive as we might have hoped for, but nonetheless, this is the first time anyone's been able to show that metabolic rhythms within human tissue and molecular rhythms can, to some degree, be regulated by meal timing. Finally, before I move on to other studies, we have some unpublished data still from this experiment. I'm going to very briefly go through this slide because there's a lot of information here. But here we looked at metabolite rhythms within the blood using our metabolomics facility. And we found, as we might have predicted, that some of these metabolic rhythms were actually delayed. And so about half of all the metabolites that we found to be rhythmic in both late and early meal conditions, about half of them were delayed by the late meals. However, some of them were not changed, and some of them actually showed a phase advance. And so very clearly, the way in which these different metabolites are responding to meal timing is very, very complicated, and we're still yet to fully understand what this means. Okay, so that's all I want to say about how we can use meal timing to reset our circadian rhythms. Now let's get on to some more nutritional physiology for you. The first thing I want to show you is some example evidence that our postprandial responses, our short-term acute meal processing biology actually changes across the day. And this was a study published by some now retired colleagues at the University of Surrey nearly 20 years ago. And this is a very simple study in which male and female human volunteers were given an identical meal either during the daytime or during the nighttime. And what we're looking at here is on the y-axis, we're looking at how much triglycerides were increased within the blood, and on the x-axis we're looking at time. So time 0 is when the participants ate their meal, and blood samples were taken for 9 hours after that meal and then analysed. The white symbols represent the data from men, and the black symbols represent the data from women. The diamond shapes represent the meal when it was given during the day, and the triangles represent the data when the meal was given at night. And the simple message here is that particularly in men, but also it seems in women as well, if a meal is eaten at night, you get a much higher peak of fat accumulation in your blood a few hours after that meal, and it takes longer for that fat to clear back down to baseline concentrations. So basically, if you eat a meal during the night, you get more fat in the blood over the few hours after that meal. And of course, concentrations of fat and lipids in the blood is one of the main cardiovascular risk factors. And so potentially this could be one of the reasons why people who habitually and regularly eat at night, such as night shift workers, are known to have a high incidence of metabolic and cardiovascular disease. There are many reasons why that may be, but one of them could be that because they're eating often in their biological night, they have over a long period of time much higher concentrations of fat within their blood than most other individuals. We can also see similar kinds of results if we look at plasma glucose after a meal. And this was a study published by colleagues at Harvard University in the USA. And they used a specialized form of protocol, which I won't explain here. But what we're looking at are two different sets of data from the same people. So this is a repeated measures experiment. And the green lines that you can see are when people are eating and sleeping in phase with their internal circadian clock. The red line is when people are essentially eating out of phase with their circadian system, so that when they're eating basically at night, at their biological night. The top graph represents changes in plasma glucose after a meal. And you can see that after their breakfast, their lunch and their dinner, when they ate those meals during the biological nighttime, there is always a higher concentration of glucose in the blood after that meal compared to when they ate it during the biological daytime. And that was true for all 3 meals. And not only was there a higher concentration after the meal, it takes longer to come back down to baseline. And again, having prolonged high levels of glucose, of sugar within the blood, is again obviously something that we would like to avoid in terms of long-term health. And these changes in glucose occurred despite there being any decrease in insulin secretion in relation to that meal. In fact, actually, it was found that for lunch and dinner, there was an increase in insulin when the meal was eaten during the night compared to during the day. And so what this tells us is that, as has been suspected now for many decades, we actually have a daily rhythm of insulin sensitivity. During the morning, we are much more sensitive to insulin than during the evening and during the biological night. And this is just one way in which that has been shown by researchers around the world. So now let's move on to how we can use timed meals as a long-term dietary intervention to hopefully improve long-term metabolic health, both within the general population, but also potentially within specific patient groups. And there are many different ways in which people can use dietary interventions. And so the term chrononutrition, although we're using it here in this talk, is actually a very broad term. And there are various ways in which we can use chrononutrition interventions. And these are just some cartoon diagrams showing you how 2 of these types of interventions can be viewed. So looking at the top in panels A and B, is one type of experiment that's been published now a number of times over the past 7 or 8 years. And this is comparing the size of breakfast compared to dinner. And of course, this was mentioned in the introduction to the talk, the whole idea of eating like a king, dining like a pauper. And so in this sort of experiment, people are essentially using 2 different experimental conditions. So in panel A, we have an example of someone who is having a large breakfast, a medium-sized lunch, and a small dinner. Compared to in B, on the top right of this slide, someone who is eating meals at the same time of day, but having a small breakfast, the medium-sized lunch, but then a large dinner. So I'll show you some data from those sorts of experiments very, very soon. I will then finish by going on to a newer sort of intervention study called time-restricted feeding. And Sachin Panda, working at the Salk Institute in the USA, has done a lot of animal work to set up this field. And now many of us around the world are actually starting to try and apply that to human beings. And there are many ways in which we can use time-restricted feeding. The basic premise here is that you restrict the time between the first and the last energy intake across a day, hence time-restricted feeding. What that also means is that every day you have a longer fasting period from the evening through to the next morning. And one thing we cannot say yet about this approach is whether it's the timing of the meals itself or the longer fasting duration that's causing the biological effects. However, you could argue that if it's working in patients, that's the most important thing over the mechanism through which it works. So if we look at the bottom right of this slide in panel D, this might be someone having a typical kind of working lifestyle food pattern where they have a fairly early breakfast, possibly around about 7 or 8 o'clock in the morning. They then have their lunch around about 1 or 2 o'clock in the afternoon, and then perhaps they're having a late dinner around about 8 o'clock in the evening. So in this case, they are spreading their energy intake over at least 12 hours of the 24-hour day. And people are looking at different ways in which we can restrict that. So in panel C on the bottom left, this is a cartoon representation of what's referred to as early time-restricted feeding. And the way this works is that lunch and dinner are actually brought forward so that you eat the same meals, but you eat them in a much more condensed way. So there's a much shorter time between the breakfast and the dinner, and then there's a much longer fasting period between the dinner and the breakfast on the following day. So that's an introduction to these different methods. Now let's look at some of the data that have come from them. So 2 studies were published back in the year 2013, which first got a lot of us very interested and very excited about the idea of comparing the size of our breakfast comparing to the size of our dinners. So on the left was a study conducted in Spain, and this was designed as a calorie restriction weight loss study. But certain researchers, certain colleagues in Spain, after this study had been completed, went back to the data and split the participants into 2 groups. Those who had their main meal of the day, which in Spain is typically their lunch, those who had their lunch early, before 3 p.m, and those who had their lunch late after 3 p.m. So the early eaters, shown by the white circles, are those who had their biggest meal early. The ones shown by the black circles are the ones who had their biggest meal later in the day. And what you can see very clearly is that after about 1 month of this calorie restriction intervention, the ones who had their main meal earlier in the day showed a significantly increased amount of body weight loss compared to those who had their main meal later in the day. And you can see that by the end of the study, after 20 weeks of this dietary intervention, actually that was quite a large difference of about 2 to 3 calories on average more lost by people with the same daily calorie intake, but just having their main meal earlier on within the day. Around about the same time, a group in Israel did a similar kind of study, but this was a designed intervention to actually compare the intervention of having a large breakfast and a small dinner with a small breakfast and a large dinner. And you can see some of the data from this study on the right-hand side of the slide. So on the top right, we're looking at body weight in kilograms. The ones shown in the white circles are the participants who had a small breakfast and a large dinner. The black circles are the ones who had a large breakfast and a small dinner. Now, these participants were carefully controlled, so they had their meals at the same time of day, and they ate the same number of calories every 24 hours. It was only the distribution between breakfast and dinner that changed between the 2 groups. And you can see very clearly here that again, after about 4 weeks, those individuals who had a higher proportion of calories in breakfast actually lost weight significantly compared to those who had more of their calories in the evening for dinner time. So you could see that very clearly in body weight, but you can also look at other basic markers of these people's metabolic health, for example, by looking at their waist circumference, which is a measure of their abdominal fat, which of course, as we know, is the type of fat that is most linked to poor metabolic health. The same group in Israel have also looked at other metabolic markers. And here they used basically the same experimental condition, one with a large breakfast and a small dinner, one with a small breakfast and a large dinner. And here they looked at the overall glucose concentration across the day. And what you can see from on the left of this slide are the postprandial glucose responses. The black circles, which unfortunately doesn't come out so well here, represent the individuals with a high breakfast, and so as you'd and a small dinner. The white circles are the ones with a small breakfast and a large dinner. And as you'd expect, the people who had a large breakfast had a larger glucose rise in their blood compared to the small one. And those who had a large dinner had a much larger rise in glucose compared to those who had a small dinner. That's what you'd expect. However, if you look at the overall increase in glucose after a meal across the whole day, those who had their large breakfast and a small dinner had overall, across the whole 24-hour period, a much smaller rise in their plasma glucose than those who had the same number of calories but distributed so they had a large dinner and a small breakfast. So this is another way in which simply changing the time of your meals, or rather how large your breakfast and your dinner are, can not only affect things such as body weight regulation and adiposity, but can also potentially be important in controlling your plasma glucose concentration. And of course, this might be very important for patients with type 2 diabetes. So that's all I want to say for now about the morning versus evening calorie loading, but I'm very happy to answer questions at the end of the formal presentation. To finish off, I just want to introduce you to the concept of time-restricted feeding. I mentioned a few minutes ago that this was set up by a group within the Salk Institute in the US. And this is one study that they and others have also done, looking at time-restricted feeding within mice. And it's just a cartoon representation of what they found in this very impressive paper back in 2012. And so in this study, there were two groups of mice, which were housed on a 12-hour light, 12-hour dark cycle. The only difference between these mice was one group was able to eat their food ad libitum at any time of day in which they wanted. The other group was only given access to food during the time when the lights were switched off. And what they found was that the animals with the time-restricted feeding seemed to have much more robust 24-hour rhythms of their metabolic cycles, but they also had less adiposity. They were much more resistant to a high-fat diet. They had reduced glucose intolerance. They had reduced leptin resistance. They had improved liver physiology, so they had much less fatty liver accumulation and pathology of the liver. They had reduced inflammation, which of course is very important for many cardiovascular and metabolic diseases. And they had various other improvements in terms of their physiology as well. And what I'm going to show you on this slide is just some of the data, the actual data from this study. There are loads of data in this paper, so this is the only bit that I'm going to show you. And here we are looking at body weight change and adiposity. So on the top left of this graph, we're looking at 4 different lines. So we have animals that either had their time-restricted feeding or their ad libitum food. Some animals were given normal food, some were given high-fat food. However, in all 4 groups, all animals ate the same number of calories per day. So there was no difference in energy intake across from day to day in any of these groups. But what you did see, particularly on the animals given a high-fat diet, was a really big change in body weight and fat accumulation. So the bottom left of this slide, we're looking at the photograph of 2 mice. Both of these mice were given a high-fat diet, but the one on the left was given ad libitum access to food. So this mouse could eat its food at any time over the 24-hour period, whereas the animal labeled FT is the animal given the same diet that ate the same number of calories every day, but was only given access to food when the lights are switched off, which is the normal active time for these nocturnal animals. And so simply by changing the time at which energy was consumed, not by changing the total amount of energy consumed, these animals are very relatively resistant to a high-fat diet. And you can see that quantified on the bottom right of this slide. So time-restricted feeding seems to work very well in animals. Does it work in humans? And so my group, but also others, have started to publish some work on this. This is one small pilot study that we published in Human Volunteers a couple of years ago. A very simple study where we had small groups, only 6 or 7 people. So again, it's a very small pilot study, and they had a 10-week intervention. In one group, in the control group, these individuals were asked to eat according to the same daily pattern that they would normally do. In the time-restricted feeding group, the TRF group, they were asked to delay their first calorie intake of the day by 1.5 hours and to bring their dinner, their last calorie intake of the day, forward by 1.5 hours. So we were in effect restricting their daily feeding window by 3 hours every day. However, we didn't ask them to change what they ate. We enabled them and we allowed them to eat whatever they wanted as long as they ate it within that time frame. And giving them this flexibility, we found that the most significant change that we found was that when you actually reduce the feeding window available to these people, they actually ate significantly fewer calories, much less energy compared to the people in the control group. So in our particular study here, we cannot say that any metabolic factors we found were not controlled or not driven by this change in energy expenditure. However, if simply by restricting the feeding window is a good way of helping people reduce their daily calorie intake, then that in itself potentially is quite a valuable thing. Other individuals and other research groups have done a more controlled laboratory style intervention. And this is just one of many examples that I could pick at. And I picked this one out because it's really one of the first big human studies to be published in this area. And this is another study which looked at what they call early time-restricted feeding. So there are two groups, and in this particular case, it was a crossover design. So the experimental volunteers did both experimental patterns. In one pattern, they had a feeding window of 12 hours where they were able to eat between 8 a.m. and 8 p.m. and then they fasted for the next 12 hours. In the early time restricted feeding window, they had their first calorie intake of the day at 8 a.m. but they started their daily fast at 2 p.m. So this is quite a big restriction on their feeding window. But in this case, the calorie content was controlled so that they ate the same number of calories in the day. And again, this was a crossover control. So each participant did both different types of intervention. And without going into the data in detail, what they found was that the time-restricted feeding group showed a reduction in postprandial insulin, but an increase in insulin sensitivity, an increase in the function of their beta cells, the cells that make insulin within the pancreas. There was a decrease in blood pressure, a decrease in oxidative stress, and also a decrease in evening appetite. And of course, all these things are highly desirable in terms of improving metabolic health. They did find a slight increase in the fasting triglyceride concentration in the blood, which potentially suggests that there aren't widespread benefits in all aspects of metabolic health, but the overall picture was very much that time-restricted feeding looks to be good for improving metabolic status within these participants. Okay, so to summarize this third and largest part of the talk, chrononutrition is a single term that we use to describe the interaction between meal timing, circadian rhythms and health. And it's a new emerging but very high profile area of research. However, although it's a single term, it actually refers to a whole range of different aspects of biology, both short-term effects, such as helping us to potentially reset our circadian rhythms, which may be very beneficial to people working shifts and people who are exposed to relatively regular jet lag. It also explains some of our relatively short-term postprandial responses. So the fact that we process meals very differently in the evening and at night compared to during the day. And in simplistic terms, the metabolic response to a meal is worse during the night, and we have higher concentrations of sugar and fat in our blood after eating the same meal compared to if we ate that meal during the daytime. However, something which is potentially the most important and the most relevant in terms of practical clinical healthcare is that people are using the basic biology that I've described to you to try and devise long-term chrononutrition interventions by changing either the time at which we eat or the size of meals at different times of the day. There's evidence now building that we can actually induce long-term improvements in metabolic health, both within the general population, but also within particular patient groups, such as those with severe obesity and those with, for example, type 2 diabetes. So the very final thing I want to do is just briefly round this up and particularly emphasize to those of you with a clinical interest what I think are the strengths, the weaknesses of the field so far, and where I think we need more research to actually be able to inform us to give much more strong advice to our patients. So this is what I've tried to do on this slide. So chrononutrition, one of the important aspects of chrononutrition is that it builds upon fundamental biology, the circadian rhythms of metabolism that I mentioned in the first third of this talk. And that's important. It's not just something that people have started to do. It's something which has been built upon very, very solid biological, basic biology foundations. As I've just mentioned, there are many different types of chrononutrition intervention, which means that the field has a lot of scope for trying different ways in which we can help individuals with their body weight and their metabolic health. And it may be useful for not just for the general population, but also for specific groups such as shift workers. And shift workers are often mentioned in these sorts of talks because it's known throughout the world that up to about 20% of the working population all around the world actually do regular shift work. So this is a very large group of people in society, and it's known that shift workers have a higher incidence of things like metabolic disease, cardiovascular disease, and even other diseases such as cancers as well. So if we can use relatively simple interventions, not just changing, for example, their sleep patterns or their light exposure, but also helping them by changing their meal times, then potentially there's a very big way in which we can have a big impact on health across populations around the world. And another benefit of chrononutrition compared to some more traditional dietary interventions is that the main focus of chrononutrition is just the timing. It's well known that most diets are successful in the short term. If you reduce energy intake and or increase energy output, then of course you're going to get a short-term benefit to health. But as I'm sure many of you know much better than me, one of the big problems in terms of using dietary interventions is helping people maintain that dietary intervention. People can often lose weight, for example, if they're on a weight loss diet, but it's keeping that weight off, which is one of the most difficult things. And one of the reasons why that may be difficult is that if you restrict what people can eat and you don't allow them to eat food which they enjoy, then obviously it's going to be more difficult for them to actually keep refraining from that very pleasurable, enjoyable food over a long period of time. Because chrononutrition doesn't necessarily work by restricting food type or food quality, it's just about the timing, then this might be one advantage which will enable people to adhere to this kind of intervention over a longer time scale than they might be able to do for other sorts. And the early evidence, which I've briefly summarized for you today, is certainly encouraging. Most studies that have been published show positive benefits to these kinds of chrononutrition interventions. However, I must also point out that not all studies show benefits. The vast majorities do, but there are one or two out there which don't. But that's what you'd expect from any kind of research field. Not all studies agree. Partly because some people are using different types of intervention, also because obviously if you're studying people in different ethnic populations, it may well be that some people in certain parts of the world are more responsive to this than others. So as yet, there's a little bit of caution, but overall, the evidence, I believe, is very encouraging. And if there was one take-home message which you might be able to start to use, albeit perhaps with some caution in the clinical setting, is that I believe that there's now good enough evidence to suggest that if we have a higher proportion of our energy in the morning with a reduction of energy in the evening, that has now relatively good evidence to show that it confers some kind of metabolic benefit. Some studies don't show that, but no one has shown it, as far as I'm aware, to be adverse to people. So the overall benefit seems to be there. Also, time-restricted feeding. If people can reduce the time between their first and last energy intake of the day, there's mounting evidence to suggest that that is likely to be helpful. Again, there's one recent study which suggests that there wasn't much of an effect, but overall, the vast majority of studies suggest that this is actually something which is likely to benefit a large number of people. So with that, I would just like to acknowledge my collaborators and the researchers who have done all this work, and of course my funders as well. Hopefully that's been an introduction not just to the basic science, but to the applied science, which I hope you found useful and interesting. And I'm very happy to take questions from you for the rest of this presentation. So thank you very much.
Thank you so much, Dr. Johnston. That was indeed a wonderful presentation. So excellently you have covered right from basics to the applications of this particular concept of chrononutrition. And many of us actually even include, many of us including even academicians and even partly researchers have some apprehensions like whether we will be able to understand the topic or not. But you made it so easy for us to understand. The way how you have sequenced your presentation is really excellent. Thank you so much, Dr. Johnston. And during your talk, you have given very good ideas, so many ideas of how people can actually take care of these circadian rhythms and how, and the ambitious, one possibility that you have given is it is possible to reset the body clock. So that is something very nice, because you must be definitely aware of the adaptation process, because since long the dietary habits have been changing, and they have changed towards more towards negative habits related to high energy consumption, meal composition changes, meal timings changes, etc. Probably these could be the demands of work schedules, or these could be probably individual preferences also. Okay, but however, the negative impact of frequently changing more towards negative side, how it influences the metabolic health, that you have emphasized very, very clearly through various studies, research studies. This is what actually I was saying in the beginning, that the evidence that you have generated through your research work and you have gathered from others researchers is indeed very, very valuable. That has made it very clear that there is a very strong scientific proof beyond all these concepts that you are actually showing us. So here, shall we start the question answers?
Yes, please.
Yeah, myself, I'm also very, very eager to actually discuss certain things with you. Okay. You have mentioned that the time restricted feeding trials, okay, they have proved the need to have need to have disciplined eating schedule. In other words, okay, time restricted feeding trial, the benefits of that, you have proved, you have shown the scientific data about that. So can you just elaborate on, like, you have of course given some examples of the time gap between two meals that you can have, and during which you are elaborating more on breakfast and dinner. Okay, two meal concept actually you were more concentrating on, whereas usually nowadays what we see is multiple meals. It is not just two meals, minimum 3 meals, and there are some suggestions in certain clinical conditions for more frequent meals. So it may go up to even 6 meals, but the same energy content which has been divided into multiple meals. So under such circumstances, what do you think one can do? How can they modify the meal division, because it is very difficult to stick to just 2 meals a day?
Yeah, that's a very good point. And something that I didn't have time to talk about today is the fact that it's not just the timing, but also the frequency of meals and snacks that people have. So there's a whole wide scope of how people study chrononutrition, and some people have indeed tried to do association studies between meal frequency and metabolic health outcomes. In terms of the two kinds of approach that I talked about today, I think in terms of the relative size of morning versus a breakfast meal compared to a dinner meal, I think if you wanted to extract that to people who don't have 2 or 3 defined meals a day, I think the way in which you would do that is just to say that overall in the morning, it would be better to have more of your energy consumption, and overall in the evening to have reduced energy consumption. And whether you do that in one big morning meal and one big evening meal, or a range of snacks across the day, I think as long as you start to increase the proportion of energy in the morning and reduce it in the evening, I would guess that you would see a very similar kind of effect, whether you only have 2 or 3 big meals and nothing in between, or whether you have lots of small snacks. In terms of the time restricted feeding, Again, there are lots of different ways in which people have done this. I only showed you 2 examples, but one thing which we haven't really studied yet is there a difference between having your short duration energy intake early in the day or late in the day? It may well be that if you only consume your energy within, say, 6 hours every day, you might get a different response if those 6 hours fall within the morning or within the evening. There's evidence to suggest that whatever time of day you have that short window, it will be beneficial to you. But so far, people haven't directly really compared the two. And that's something which I've got a PhD student who's currently studying that, and I'm sure other people around the world are also looking at that. But overall, with time-restricted feeding, it seems that as long as you restrict that time between first and last energy intake, you will get some benefit, irrespective of whether you like to eat mostly in the morning or the evening. And in the basic concept of time-restricted feeding means that it shouldn't make much difference whether you are someone who has regular snacks or 2 or 3 large meals. As long as you have all your energy within that small window, the evidence so far suggests that you are likely to have a metabolic benefit.
Okay, so that is where actually the individualized or personalized nutritional intervention plan, it comes into place. Because according to individuals' dietary habits, probably you will be able to personalize the suggestion while following this kind of regulations, the circadian rhythm. You can follow the, you can plan the chrono-nutrition intervention according to personal preferences as well, with mild modification in the timings.
That's something which I think we ultimately want to get towards. Partly due to behavior. One question that we haven't talked about here, but we've recently done some work on, is actually looking at people's decision making when it comes to their behavior, because it's very easy for someone like me to do an experiment and show that if you have an intervention, it will benefit your health. But of course, if people can't actually use that intervention in their normal day-to-day life, then it's simply a scientific finding. It's not something that we can use for people and for patients. So one thing that I've been interested in, and I know others are as well, is actually finding out what are people's sociological influences on their behavior. One thing, for example, that is likely to be an important factor is family life and childcare. People often like to eat. Eating is a social behavior. People generally like to eat as a family. And of course, the age at which if you have children, the age of your children might affect what time you would prefer to eat. So there are a whole range of different things like that. So yes, it needs to be individualized, and looking at people's daily behavior, their work schedule, their family life is all another part of how we need to think about using this basic science and actually using it to help people in their real life.
Yes, that is very right, because so many other variables are there that might influence the way how your body reacts to the food consumption. So I will just pose one last question before I invite Mr. Shekhar into the discussion. This is like, you have proved beyond doubt that large meal at night is metabolically risky. Okay, you have proved and you have shown the scientific evidence related to that. So here, is it only the timing, like a night dinner? So that is large dinner, which is metabolically risky, or what about the factor of meal composition over there? Because you have also mentioned that there is a tendency of eating calorie-rich food at dinner timings. So the timing versus meal composition. Can you please throw some light on that?
Yes, again, that's a good point. And again, it shows the complexity of what we're talking about here. When people have done focused experiments, usually what we do is we try to have as few changes in our experiments as possible. So when we do an experiment to particularly look at a morning versus an evening meal, we would usually make sure we give exactly the same meal composition. So that way it makes it more easy to interpret the results. But yes, on top of that is also the fact that, as I mentioned and you've just mentioned, that in the evening, and when people are tired, they tend to choose energy-dense food. So I suspect that in real life, you've got two things, both of which are probably not good for your metabolic health, that will add to each other. Just by eating at night, you get a higher rise in sugar and fat in your blood after the meal. But if you were also eating an unhealthy, energy-dense food, that's likely to make the effect even larger. So I don't think anyone has really directly compared morning versus evening with different macronutrient content. But my guess would be that if you eat, if you like, a poorer quality food at night, you'd see an even bigger difference between your morning and your evening responses.
Okay. So we need to still generate data on that.
Yes, I think so.
Generate data. So now I will invite Shaker to.
Thank you, Subhadra. And I have to mention here, Dr. Johnston, that was one of the most amazing presentations I've ever heard. And for the nutrition fraternity here, you are like a god who's coming and telling us meal timings are also equally important. And as a fraternity of nutrition, I would like to ask you, what are the kind of studies that we as nutritionists or dietitians can do, because we do not generally have too much of infrastructure like you. So can you suggest some very simple experiments that we can do in our colleges or in wherever we are in our hospital setups? Thank you.
Yes, certainly. So thank you very much for those kind words. It's very nice of you. I should very much acknowledge many colleagues around the world who've done far more impressive work than I have, but it's nice to be part of the field. So in terms of experiments, I think that the two kinds of intervention that I mentioned towards the end of the talk are the ones which, for those of you who are clinical nutritionists and dietitians, are the ones which I think are most relevant to your work and your practice and to your patients, because ultimately we want to use basic biology to help improve healthcare within all sorts of people. So these sorts of experiments can be relatively simple to do. Because they tend to last over a long period of time, they can be done with participants while they're living their normal life. Of course, when people are living their normal life, you can't check so easily that they're doing what you ask them to do. You have to trust what they do, but that's just part of doing real life, if you like, in the field experiments. So you could potentially have some of your patients and ask them to change their meal times, and that's something which should be relatively simple to do. In terms of what you can measure, well, obviously things like body weight and waist circumference as the marker of abdominal fat, which is, as you know, is the unhealthy form of fat. So you've got very simple measures that you can take there. Fasting blood samples, for example, if a patient comes into a clinic, particularly in the morning, you could take a fasting blood sample and measure markers such as lipids or glucose within that. One thing, of course, that is important to me as a circadian biologist is if you do take a blood sample from a patient, the time of day at which you take that sample is going to have a big effect on what you measure. So if a patient comes into your clinic in the morning or in the afternoon, even if they're fasting, you will get your natural circadian rhythms mean that what you measure is going to change between morning and evening. So if you do that kind of study, one thing that you need to be very wary of is that if you take a sample from someone, you measure it at the same time of day. And I imagine for most people, that would be easiest in the morning after an overnight fast. And that way you're controlling time of day, and also you are taking out any effect of an acute big meal. Something else that you could measure, particularly if you're interested in glucose control, for example, in diabetes patients, is to measure something like HbA1c, which is a long-term marker of glucose control. So even if you are unable to control the immediate glucose concentration, if you measure HbA1c, you get a factor of glucose control over the previous weeks, which potentially is actually more relevant to someone's long-term metabolic health. So there are things like that that you can do, which are relatively easy, don't require too much equipment, and don't require too much in the way of cost.
So the next question is, there are lots of questions from our Indian dietary point of view, because we also believe in Ayurveda. I don't think you might be knowing much about it, but we have some foods for Prakriti, Vikruti. So there are a lot of questions coming on those areas, but I'm sure what you have said must be making some relevance to these, because we also follow practices of having just 2 meals a day, and they do really help, because our grandmothers have been using those kind of diet plans earlier, and they even do something called a complete fast just to detoxify themselves. So I think most of the audience, I'm not going to be asking questions regarding Ayurveda, and so many questions being asked here. But sir, can you tell us can you suggest some books for reading on circadian rhythm and chrononutrition? Are there any nice books available for us to update ourselves, apart from journals? Yeah.
Yes, so there are some books that have come out quite recently. Circadian rhythms is still quite a new area of science, but there have been some that have come out. I mentioned a colleague in the USA called Sachin Panda, who's recently produced a very nice book on circadian rhythms and metabolism. And as I mentioned, he's personally done a lot of his work in animal biology, but he's now starting to do some human studies as well. But he has really been at the forefront of some of this chrononutrition work at the basic science level. And he does a lot of work in terms of publicizing it to the media and to the public as well. So that's one book which would, I think, be probably of most relevance to many of you. By the way, if there's any information that you would like after this talk, please feel free to email me some of these questions after we finish the cast, and I'll email you back some answers which you can share. Okay. And one of the things I can do is actually just have a little hunt around for some more books which may be relevant, and I can send you titles of that as well.
Thank you so much for that. And there are a few questions, but most of the things you have already answered. There are a lot of questions where diabetes is concerned, and how can this be fitting to treat people in type 1 and type 2 diabetes is one of the questions that so many students have asked. So I don't mind even if you could answer these kind of questions. We'll definitely put these questions on, and you can answer them a little later. But these are the major questions are that how does intermittent fasting and chrononutrition help, and intermittent fasting also talks about the same thing what you just now said, that it's just 2 meals a day and then you have a long gap between 2 meals. So it is also being worked on diabetic patients here in India. And many people are trying these diets. And there are a lot of opinions, of course. The lobby is for and against. And so many of these nutritionists do have a lot of queries in their brain that, whether it really works or no. But I think with your studies, it does emphasize that there is definitely an answer coming for an intermittent fasting here. So I think a lot of studies need to be done in Indian context, because our seasonal variations are there. The day and night timings are a little different from other continents. But there are some continents where there is not much of daylight. It's mostly night time. So in those cases, does mealtime really help? Especially in areas where there's too much of night time and very few hours of sunlight available.
So the whole idea of kind of seasonal changes in humans is in itself a really big and interesting question. And before I started doing my current research, I actually studied seasonal biology up in the north of Scotland and places like that, where you do get these really big changes in day and night patterns. There is some evidence that there is some kind of seasonal biology within humans. So, for example, in controlled laboratory studies, people have shown that day length, so you obviously have a long day and a short night in the summer, and the opposite in the winter time, at least in the northern hemisphere. And there's some evidence that some of our hormonal markers might be sensitive to day length, although much more subtly than a lot of animals that live in the wild. There's some evidence, for example, if you go before industrialization, that there are rhythms in birth rates, whether that's to do with rhythms in our reproductive physiology or rhythms in behavior, then it's difficult to say. So in general, it's quite difficult, I think, but certainly at the moment, to show strong rhythms in metabolism in people. I suspect that the bigger control will be in people's behavior. If you're living, I don't know, say in a polar region of the world where it's dark pretty much 24 hours a day during the winter, I imagine how that affects your behavior, and therefore how it affects your mood, how it affects your eating, is probably going to be a bigger factor than the actual seasonal biology within that person.
Thank you so much for that answer. And Subhadra, do you have any question or should? Yeah.
Yes. No. I just want to ask one or two questions, Dr. Johnston. Can I?
Yes, please.
Yeah. There is one very interesting question. How to incorporate exercise with respect to chrononutrition? Because now most of the dietary interventions are along with exercise guidelines as well. So whether it will have a cumulative benefit or do they interfere with each other?
That's a very interesting question. And I must admit, I don't know. I don't feel I can give you a definite answer there. I know that there are colleagues in the exercise physiology field and the nutrition field who are looking at how the timing of exercise relative to meals can help. So I know, for example, colleagues at the University of Bath who I've collaborated a little bit with, and they are very interested in this. And I'm sure there are people all around the world doing that. Unfortunately, I must admit that I don't know the answer to that myself. I imagine that the combination of the two will have some benefit, but I suspect that they are unlikely to, if you like, cancel each other out. I imagine that you've got two things that are beneficial, which will probably add benefit to each other. Whether the relevant timing of meals and exercise will change how they benefit each other, that I don't know, I'm afraid.
Sometimes some of these questions actually intrigue interest in us also, and in scientists like you, probably you may come up with another research idea, another
yes, definitely.
That will have. And another question actually is like when we want to follow these chrononutrition principles in clinical conditions, okay, while following the dietary guidelines which are already prescribed for a particular clinical condition besides diabetes, then do you have any information to share with us, like how we can modulate these chrononutrition principles for various clinical conditions, or is that also a field that requires more and more research?
I think that, to be honest, will require a little bit more research before we have clear evidence. Most of the work that's been done so far is in relatively healthy volunteers, because that's the way we start. We start looking at the basic biology, then we apply it to kind of healthy, normal people, if you like, and then we start to apply it to patients. And at the moment, most of the work has been done in relatively healthy people, but there are groups who are starting to look at applying this to type 2 diabetes is one of the obvious examples, but also people with severe obesity. So that data is starting to come out. My guess would be that the sorts of improvements that I've described to you would apply to most patient groups as well as to the broader general population. However, that's only, if you like, an educated guess at the moment. I strongly suspect that over the next few years, we'll have a lot more data coming out where people have used these approaches in specific clinical populations. So hopefully in a few years' time, there will be a very clear answer to that question. At the moment, I suspect there will be similar benefits, but that there's a lack of evidence at the moment, I think.
Yes. So actually the questions also give us more scope to research more and more, and or search for more information and come up with certain answers for that. But your talk is really thought-provoking, Dr. Johnston. I now invite Mr. Shekhar to formally thank you on behalf of the Nutrition Society of India Mumbai chapter. Thank you so much.
I have been given the privilege to give a vote of thanks, and I'm really, really indebted to Dr. Johnston. Sir, namaste from India. Thank you so much for accepting our invitation and being here in time and doing a wonderful job with your presentation, taking all kinds of questions that we have posed on you. Thank you once again from all of us from NSI Mumbai chapter. I would like to even thank Kellogg's India. And here, Kellogg's is definitely into breakfast cereals. And I'm sure Dr. Johnston has highlighted that a morning breakfast is a must. And I think Kellogg's stands by that particular vision and mission statement. And I thank Kellogg's to give us this particular platform and all of us to listen to such a beautiful deliberations on chrononutrition, which is something very new. It is in its infancy, but we are definitely giving you medicine to take home. And I think there are a lot of take-home messages. And I hope all the nutritionists and the dietitians who are on this particular platform are having some wonderful ideas in their brain to start pursuing some great research in this line. And thank you to Subhadra, my co-partner in this. And she has been instrumental in getting in touch with Dr. Johnston and giving this beautiful baby for all of us to enjoy today. And to my SM team, the young, beautiful girls, Chandani, Aisha, and Neha, for really helping us out without any hitch, having a platform like this and connecting all of us together. Thank you so much, you wonderful girls. And thank you once again, Johnston, Dr. Johnston. We'll definitely keep in touch and we'll definitely see you somewhere. Thank you so very much from all of us from India. Thank you.
Well, thank you very much again for those kind words. It's been a pleasure talking to you. I hope it's useful to you, and perhaps we can update you sometime in the future when we have a little bit more information. Thank you very much.
It's been wonderful.
Very happy if you will be able to, if you can share that information, whatever new information you are generating with us, so that we can dissipate that information to all our members and all the participants of our webinars. We have a huge number of participants for our webinars, and we have been organizing them right through the lockdown period. And we have the list of database of so many of them. So we will be able to share that information because it's going to be very useful for all the nutritionists across the globe. Thank you so much.
Excellent, I hope so. Okay, thank you again.
Thank you. I now invite Aisha to make certain announcements about certificates and other things related to this webinar. Aisha, over to you.
Thank you so much, ma'am. And thank you so much, Dr. Johnston, for that very interesting presentation. Before we officially close, I have a few announcements for the participants regarding issuing of certificates. Just give me a moment while I share my screen. The participants will receive the certificate for this particular webinar once they successfully fill the feedback form, the link for which has been sent to all registered participants on the night of 11th December. You will be able to fill the form only after 6 p.m. today, and the link will remain active till midnight tomorrow. So we request all participants to kindly fill the feedback form before midnight tomorrow. The certificate will be sent to all participants only by 25th of December 2020. We therefore request you not to email us asking about the certificate prior to this date. Please download and save the certificate from the Google Drive link that you will receive in your email. Also, this session will be available for viewing later on YouTube, and it can also be accessed using the YouTube link that has been mailed to all the registered participants. We thank you again for your active participation and patient listening. Thank you so much and have a good day.